Thank you for the invitation to present probably the first results from the BAT1K project. And I would like to make sure at the beginning that I mentioned this is a great team effort that involved a number of people with Emma Thieling, Sonja Wörnes, Jean Myers, and myself being kind of the major PIs involved in this current project. So why studying bats? Why we are interested in that group of species? So first, there are lots of them. 20% of all living mammals are bats. There are about 1,400 different species. Bats are the only group that evolved powered flight. Many bats can use echolocation to hunt and navigate in complete darkness, like in caves, for example. Many bats are able to learn vocalizations, which they use for social interactions and communications. It is also becoming clear that bats are reservoirs for some of the deadliest viral diseases like Ebola or SARS. But bats are able to tolerate and survive these infections. And there's evidence that they possess a special immune system that is able to dampen and resolve the inflammatory immune response much better than mice and probably other mammals, including probably us, can do. And finally, bats live on average 3.5 times as long as non-flying mammals. And there are some species which live, after correcting for body size, even longer than human. Nevertheless, they show pretty much low rates of cancer, little senescence, so they are models for healthy aging and studying cancer resistance. In the BAT1K project, our goals are to generate reference quality assemblies eventually for all living bat species. And we'd like to use those genomes to resolve the phylogeny of bats, to get insights into their evolutionary history, discover the molecular and genomic basis of their adaptations, and use those genomes to help in conservation efforts. So I'll be presenting results from the pilot project where we generated assemblies for these 6 lovely creatures here. And these 6 bats belong to 5 different families, and they span the 2 major suborders, which are Yangocharoptera and Yinterochroptera here. Most of the sequencing and assembly was done in Dresden by Martin Pippel, Silke Winkler, and Jean Myers. I will briefly walk over kind of the procedure, how we assembled. We generated first PacBio long sequencing reads, aiming at something like 60x coverage, and this is the coverage we got after the final assembly. Because assembly size can vary, we, in a way, post hoc had something between 50 and 70x. The read length, as you can see here, is quite good. So something N50 read length between 15 and 25 kilobases. And of course, as you know, the idea is that now the reads are typically longer than the repeats in the genome, so you can assemble over them. And I also have a slide pretty much showing this puzzle analogy, right? So now you're solving a puzzle with larger pieces. That makes it easier. The assembler we used is called Damar, and that will come up on GitHub soon. And it's pretty much an improved version of the Marvel assembler that we used to assemble the axolotl genome, I think 2 years ago by now. Then we generated 10x Illumina read cloud data, which you can use for scaffolding, but we didn't really manage to generate this data for really long molecules. So it had little scaffolding information for us. So we only used those linked reads shown here to map them to the primary contigs and correct base errors. Third, we generated optical maps, and this is the N50 kind of molecule length. So you see there's ranges between 190 and 335 kilobases. And using the Solve software from Bionano, we, in a way, those molecules can be assembled in a genome map, which then gave us, in a way, the first round of scaffolds. And finally, we generated high-C data to further scaffold the scaffolds we already had, which then gave us, in a way, the final scaffolds. That was the procedure. And this plot shows in a way, the N50 plot. So this is the N50 line here, and the N50 values shown in the y-axis. Our N50 values for the 6 bats range between 10 and 22 megabases, and they are shown here in color. Then you see this dashed line here. This is the genome of the Eonycterus bat, which was produced something like a year ago with 80x PAG coverage. As expected, also has very long contig N50s. This is dashed line is a hybrid where both long and short reads were used, and all the dotted lines here are Illumina-based assemblies. And you can see, and as is expected, right, that our assemblies are something like 2 orders of magnitude more contiguous. Scaffold N50 for the 6 bats varied between 80 and 170 megabases, and this is also better than pretty much any other bat assembly out there to date. And we estimate that between 87 and 99% of each of those assembly is assigned to chromosome level scaffolds. The genome size of bats is typically a little shorter than the size of other mammals, so it kind of varies between around 2 gigabases. Nevertheless, I found it surprising that there's still quite some size variation. For example, Molossus has a genome of 2.3 gig, and this is actually almost 500 megabases larger than the genome of Pipistrellus. This smaller genome size correlates with an overall lower repeat content, which is shown here if you compare this to human, mouse, and a bunch of other mammals. Otherwise, if you look at the different types of repeats, and this is work by David Ray, this looks fairly normal with with line elements making up the largest portion of the repetitive genome. Maybe what's interesting is that 2 of these bats have a substantial portion of these rolling circle transposons, which you rarely see in the other species we looked at. And David Ray also looked at the proportion of the genome that consists of very recent transposon insertion. And here we see 2 things. First, this is very dynamic because you see that different classes of transposons are active in these different species, or have been recently active. And second, the activity doesn't really correlate with genome size. If you look at Rosettus, there's barely any recent transposon activity that we could detect. And this has actually a slightly larger genome than the Pipistrellus genome, where about 3% of the genome consists of recently inserted transposons. We use Busco applied to the whole genome assembly to estimate how complete is our data in terms of genes. And we found that something like between 93 and 96% of all the mammalian Busco genes are present in the assembly. If you compare this to the statistics of other assemblies, that looks pretty much the same. I'll come back to this plot in a minute. And then we were interested in assessing also completeness of the assembly in the non-genic regions. And here, David Jebb, who is a very talented postdoc in my lab, determined how many non-exonic ultra-conserved elements align in those genomes at an identity threshold of 85%. And the idea is that these ultra-conserved elements are kind of the tip of the conservation iceberg, and are typically highly identical across mammals. So we should detect all of them if we ask for 85% identity. What we found was that 4 of these ultra-conserved elements were not detected at this threshold in 2 of our bats. And this looks similar to this previous short read-based bat assembly, or some UCEs also missing in the dog, the cat, and the cow assembly. So this, of course, troubled us, and we investigated what's going on. And let me first show you why those previous assemblies are missing some UCEs. This is UCE 157, where we found no trace in the FELCAT8 assembly of the cat. And the reason for this is that this locus here, where you would expect to find this UCE, this overlaps a large assembly gap. And indeed, if we look at the most recent version of the cat, which was built using PacBio long reads, you see that this entire gap is closed and the UCE is actually present. So it tells us that this UCE is not lost or deleted. It was simply not present in the previous cat assembly. And our analysis showed that all these cases here are missing from those assemblies because they overlap assembly gaps, so those assemblies are incomplete at those loci. So what's going on with those 4 elements here? And here we see first, we find them in the genome. They do align, just not at 85% identity. And this is shown here. You see 5 of these bats have a pretty nice alignment to the human UCE sequence. But when you look at Pipistrellus, there are a number of mutations and smaller deletions. And of course, this initially looks like a whole bunch of uncorrected base errors, right? But we found, we aligned the raw PAG reads or Illumina reads. We found strong support for this sequence here. And furthermore, David looked at another assembly of another Pipistrellus species recently released by the Broad. And here we see that pretty much most of these mutations are shared between these 2 closely related species. So this tells us those are not sequencing errors or base errors. This is a real sequence divergence. And I should say I've never seen a case where UCE truly has that extent of sequence divergence. So it turns out that all the 4 UCEs we didn't detect at 85% identity, or the reason for this is that there is true sequence divergence. And so from this, we can conclude that our assemblies also have quite a high completeness in the non-genic regions. Then we annotated genes, and here David, in a way, was using an approach that integrates as many evidences as you can kind of collect. So briefly, we aligned genes annotated in other species to our assemblies. We used genome alignments to project genes from human and mouse, where they are obviously very well annotated, to our bats. And this is, in general, a very powerful and accurate approach. We did de novo gene prediction with Augustus, using both the single genome, so ab initio mode, and also the comparative mode, where you feed a multiple genome alignment to Augustus. And we generated transcriptome evidence using both RNA-seq to get the coverage, and also some iso-seq data to, in a way, get full-length transcripts. All this data was integrated in the consensus gene set with evidence modeler. And then our inspection showed that not every gene and definitely not every isoform is actually represented here. So what we did was copy-pasting high-confidence isoforms that we deemed to be full-length. So those coming from assembled RNA-seq or iso-seq data, but also those where both, in a way, the human and the mouse gene was projected in its entirety. So we copy-pasted missing isoforms into the final gene set. With this strategy, we annotated between 19 and 21,000 coding genes in these 6 bats. And then we investigated how complete is now the gene annotation. And here we see that at least 99.3% of all these Busco mammalian genes are completely present in our gene annotation. And this is a completeness that is actually higher than the annotations of dog, cat, horse, cow, and pig. And this completeness is only surpassed by the annotations of human and mouse, where, of course, a large community, right, provided extensive manual curation over time. And a point I would like to make is that every gene you find in your gene annotation is, of course, also present in the assembly. So you would expect that if you apply Busco to the whole genome or to the gene annotation, meaning the translated proteins, that you get identical results. And if you look at those 2 plots, this is not the case, telling us that, of course, finding gene models in the whole genome is more difficult. And that means applying Busco to the whole genome will underestimate the completeness of the assembly, right? I always wondered that why is it that human is actually missing 2% of the genes, right? They are not missing, just Busco fails to detect them. That's probably something to keep in mind. Okay, and then, of course, the main motivation of the project is to try to investigate what are the genomic determinants of these adaptations in bats. And here David used 3 different genome-wide screens to look at differences in genes. And the first screen was to look for positive selection in genes. We've been using a tool that we call TOGA, which stands for Tool to Infer Orthologs from Genome Alignments. And as the name implies, it's an unpublished method we're about to finish in the lab that uses genome alignments to infer one-to-one orthologs across a wide range of species. And with TOGA, we were able to get in the order of 13,000 genes, which we think are clear one-to-one orthologs across 48 mammals. We used HiFi to screen this large set for signatures of positive selection, and used PAML to confirm the pattern of positive selection. So 2 methods agreed on this is a gene under significant positive selection. And after correcting for multiple testing, we obtained 9 genes. So this number is probably low because this is a very conservative approach of looking for selection. And second, the branch we're looking at is quite short, right? So you would not expect to find loads of genes that are selected. Nevertheless, obviously, the downside is of having such a comprehensive set of orthologs is that you have the burden of multiple test correction. So Graham Hughes investigated what happens if we would focus only on a set of candidate genes that could be relevant for longevity and immunity. And then at the same FDR cutoff, but now having to correct for less tests, we find selection in 10 additional genes, so 19 genes in general to look at. Interestingly, 2 of the genes that came out in the genome-wide screen have hearing-related functions. So LRP2 and SERPINB6 are both expressed in the cochlea, also in other places, and both genes are associated with human disorders that involve deafness as one of the phenotypes. And this is interesting because among all the different families of bats, theropodids cannot echolocate. And if you look at this phylogenetic tree here, then there are 2 possibilities. Either echolocation evolved twice, once here and once here, or echolocation evolved in the ancestor and was later lost in this family. The signal of selection that we're detecting, that comes from this particular branch. So if the selection is to improve hearing function in these genes, and this is something that I think we need experiments to figure that out, this would then provide, in a way, molecular evidence for an ancestral origin of echolocation, and that echolocation was later lost in theropodids. And then we see that out of these 19 genes, we find a number of genes that have immune-related functions. For example, we find selection in a chemokine, in a neuropeptide receptor that induces inflammatory responses in macrophages. Find selection in IL-1 beta, which is a cytokine that activates NF-kappa beta, and in a gene called INAVA, which amplifies interleukin-induced inflammation, and it does so by also enhancing NF-kappa beta signaling. So we think that changes in these genes, in particular these NF-kappa beta modifiers, could be involved in immune system changes in bats. The second screen David looked at or performed was to screen systematically for inactivated or lost genes. And this is motivated by previous work from the lab, where we found probably a number of examples suggesting that sometimes in evolution, it can be beneficial to lose actually a gene. And that gene loss could contribute to adaptive evolution in different mammals, including those fruit bats here. So when I say gene loss, I mean that we're going to screen for stop codon, frame shift, splice site mutations, and also complete deletions of exons or genes. And if you look at this picture here, this appears to be a straightforward problem, right? If we have this alignment, we can all do this manually by hand. But if you want to do this across lots of species, genome-wide, and you want to achieve a high accuracy, it turns out that there are a number of issues that are related to the quality of the assemblies, the accuracy of the genome alignment which we're using, and also the fact that conserved genes can still change or evolve their exon-intron structure. So a former postdoc, Virag Sharma in the lab, developed a pipeline that tries to address all these issues here. And he could show that if we look at a large set of truly conserved genes, so those are annotated one-to-one orthologs between human and 4 other mammals, we get a specificity of at least 99.7%. So we think the approach has an accuracy that's high and good enough for genome-wide application. David used this approach to screen for genes lost in all our 6 bats, and we found 10 of these genes. And interestingly, 2 of those genes, again, have immune stimulating and pro-inflammatory functions. One of them is an interleukin-1 family member. The second is LRC70, which is a broadly expressed gene that amplifies cellular responses to cytokines and is able to amplify NF-kappa beta activation. And this plot is just to show that we manually validated the loss of these genes. In particular, we can show that the frame shifts and other inactivating mutations are typically shared with the assemblies of other bats. So we're sure those genes are truly lost. And if you now integrate the results for screening for selection or screening for gene loss, then we found 4 genes that appear to affect, at the end of the day, NF-kappa beta signaling. So our prediction is that alterations in NF-kappa beta signaling could contribute to immune system changes in bats. The third screen David performed was to look for systematic significant expansions or contractions of gene families. Of course, for this, having highly accurate and complete assemblies and annotations is very useful. We found 35 significant expansions or contractions, and the one I would like to highlight is an expansion of ApoBec3 proteins. And here we think that the sister species of bats, represented here by cow or dog, typically had something like 2 or 3 of those genes. And we think that in the ancestral bat lineage, some expansion, probably to 4 genes already happened. And this was then later, so later in different lineages, additional lineage-specific amplifications of these genes. In meiosis, actually, they created a second locus where we find many of these genes in the assembly. What's interesting about ApoBec genes is that they have DNA and RNA editing capacity and are implicated in restricting viral infections, so a link to immune system, and limiting transposon activity, potentially contributing to lower transposon content in bats. So to summarize, we generated, or we combined PacBio, 10x, BioNano optical map, and Hi-C data to generate reference quality assemblies of 6 different bat species. We generated highly complete gene annotations. We found selection in hearing-related genes in the ancestor of bats. And if, I'd say, experiments confirm this is selection somehow linked to hearing, then this, I think, would provide evidence that echolocation evolved once in the ancestor of bats. And then we performed genome-wide screens that look for gene loss, gain, and selection. And we found patterns of positive selection in immunity-related genes, some of them regulating NF-kappa beta signaling. We found losses of genes that would stimulate the immune system and enhance have pro-inflammatory functions. And we found this expansion of these antiviral ApoBec3 genes, as I just mentioned. There are additional analyses that this group of people performed that I don't have time to talk to you about, but I'll briefly summarize it. So we looked at changes in non-coding RNAs and microRNAs and found interesting gains and losses, and also alterations in the seed sequence of microRNAs that is predicted, and we have some experimental evidence for this, to alter actually the target spectrum, so the target genes regulated by this particular microRNA. Aris and others looked at genomic integrations of endogenous retroviruses, where we found that different types of viruses, in a way, left their footprint in in bat genomes. And a large team looked at very comprehensive genomic data of genes and non-coding regions to infer what is the phylogenetic position of bats within Laurasia theria. All this is, you can read about all this in the preprint that is on bioRxiv. And if you want to look at the data, use those annotations, look at those genomes, we set up a publicly available genome browser at the Genome Public PKS MPGDE, where you see all these, this is our final, in a way, gene annotation, and you see all the evidences we had together with genome alignments. And last slide, what we're working on the project right now is to generate a reference quality assembly of at least one representative for each of those 21 recognized bat families at the moment. And of course, we aim for having at least the same quality as we are having right now. We would like to generate, again, highly complete annotations. And of course, this data set of one representative for each family will then enable a large variety of analysis that hopefully shed additional light on cool adaptations and evolutionary history of bats. And with this, I would like to thank you for your attention. I'm happy to take questions.